Welcome, Brookings Bio students. This is Mrs. Rydell, and today we're going to look at figuring out the probability of offspring in specific crosses by doing Punnett squares. And so, in order to do that, we're gonna look at some vocab words, right? And look at how is it that I could express or show what the probability of particular offspring are going to be. And so, what we're going to do is talk about this word a little bit: probability. Probability is the likelihood that something's going to happen, right? An event will occur. And so, for example, if I flipped a coin, I could tell you a probability of what's the chance that I would get heads on the flip, or what's the chance I would get tails on the flip. And so I can write probability or express probability in a couple different ways. One way to do it is to write it as a fraction. So, for example, if you look at these three red circles and one white circle, if I put them on little pieces of paper and I put them into a paper bag and I ask you to pick out one of the pieces of paper, and I said, what would be the probability that of those four little pieces of paper in the bag that you would pick the white one? I could express that as a fraction, right? I could say I have a 1 in 4 chance of picking the white little circle, right? There's 4 pieces of paper in the bag, and 1 out of those 4 is white, right? I could do it as a percent. I could say 25%. If I divide 1 by 4, that's what I get is a 0.25, and if I express it as a percentage, 25% of the pieces of paper, the little circles in the bag, are white ones, right? I could also do it as a ratio, when we talk about ratios. So out of the 4 there, 1 of them is white and 3 of them are red, right? There's 1 white one for every 3 red ones. So I could express it as a ratio. And when we do Punnett squares and look at probabilities, we're going to use all 3 of those ways of expressing probability when we do Punnetts. So think about, if I flipped a coin, heads and tails, right? I have 2 possible outcomes to that flip. When I flip my quarter, I could have a chance of landing on heads, I have a chance of landing on tails. So if I was to express that as a probability, if I did it as a percent, right, I would say that half the time I'm going to get heads and half the time I'm going to get tails. I could do it as a fraction. I could do it as a percentage, a 50% chance of landing heads or tails, or as a ratio. Here's where you got to be careful, because the ratio numbers are not the same as the fraction numbers. 1 out of 2, 1/2 of the time I could get heads, but I have a 1 chance head for every 1 chance tails. If I want to write it as a ratio, right, I put the number for this choice on this side of the colon and the number for this choice on the other side of the colon. And notice 1 to 1, not 1 to 2 ratio. I have 1 chance of getting heads for every 1 chance of getting tails. And if you think about it, remember we have 2 allele choices. We have 2 copies of every chromosome in each body cell. So when I do segregation and I split my homologous partners up, right, randomly, alleles are going to separate. I kind of, just like a coin, I have a 50-50 chance of getting one gene choice or the other gene choice. So we can use probabilities not only to predict coin flips, we can use it to predict genetic crosses. So if you think about probability, a couple things to remember. Remember, every time I flip the coin, it doesn't really matter how many flips I've had before. The flips from the past outcomes don't affect future ones. So if I flip a coin and I get heads, does that mean next time I'm going to for sure get tails because 50-50? No, it doesn't. I could flip another head in a second. I could get 2 heads in a row. The probability of the past flip does not affect future ones. So the next coin flip, I still have a 50-50 chance of getting heads or tails, right? My past flips don't affect future outcomes. Secondly, probability works best if you're in predicting things if you're dealing with large numbers. So for example, if I flip a coin 10 times, I could get a 5 heads and 5 tails, 50-50, right? That's the probability. Is it possible I could get a 6 heads and 4 tails? Yeah. 7 heads and 3 tails? It's possible, right? It's even possible that I could flip 10 heads in a row and no tails. But that's a pretty small number, 10 flips. If I did 1,000,000 flips or 2,000,000 flips, what's the chance that they'd all be heads or all be tails? That's a pretty slim probability. The more you're counting, the larger the number, the better you're going to be, the closer your results are going to be that close to that 50-50 expected, what my expected average is going to be. Okay, in order to do Punnetts, we need a way to represent our gene choices. And remember, in our last slideshow, we talked about dominant and recessive allele choices. Talls and shorts, wrinkly, round, yellow, green. So we're going to, when we do a Punnett square now, we're going to represent our dominant allele choice with a capital letter. And usually we pick the first letter of the trait, T for tall, Y for yellow, for example. Okay? The recessive choice is going to be represented by a lowercase letter. And so, for example, if I wanted to talk about a tall, short cross, crossing a tall plant with a short plant, I could use T to represent tall. That's the dominant one. Tallness is going to show even if the recessive choice is there. And short now, my letter for short, notice I got to use the same letter. So I don't use S for short. I use little t. And that tells me, when I see those two letters together, that tells me that those two choices go together for the same gene. Size gene. I could have a tall choice, I could have a short choice. They go together. If I use a different letter, then I'm talking about a totally different gene. So keeping the letter the same is important to helping me know how many genes are involved in my cross. Okay, the other words that we need to know about to do a Punnett square are these two words, homozygous and heterozygous. If I have both alleles in my pair are the same, we say the organism is homozygous or pure breeding. Pure breeding. Think about, remember, you have two of each kind of chromosomes, so you could have both dad and mom give you the same choice, allele choice for a trait. We would say you are homozygous or pure breeding. So if I express that in letters now, two capital T's, two big T's for tallness would be what we call homozygous, pure breeding tall, though that plant is always going to make tall offspring. Lowercase t, two little t's, this is what we call a pure breeding short plant. It's always going to make short offspring. It only has short alleles to give. And so we would call both of those organisms homozygous. If my allele choices in my pair of chromosomes are different, we would say the organism is heterozygous or hybrid. Hybrid. And allele choices that are different, one big T, one little t, one dominant, one recessive allele choice, we would write that as big T, little t. Okay, heterozygous, two different letters. Remember, each organism has two of each gene. You're going to get two gene choices on every gene that you have because you have two homologous chromosomes. Okay, the other words that we need to know about to do a Punnett square have to do with what kind of gene combinations we're going to get in our offspring and what are they going to look like. So the genetic makeup, what genes they have, is called their genotype. Look at the word. It kind of says it in the word. What type of genes you have. Do you have a big allele, dominant allele? Do you have a recessive allele? Do you have two of the same? Do you have two different ones? That's your genotype. What's your genetic makeup? Your phenotype is the way you look. So if I have two dominant tall genes, I'm going to look tall. My genetic makeup would be big T, big T. My phenotype is going to be tall. The way you look, what genes you have. And both of those words are going to come into play when we start doing Punnett squares. Okay, so let's talk about setting up a Punnett square. We're going to start with a simple one gene cross. We're going to look at one gene at a time. And to do that, it's called a monohybrid cross. Mono means one. We're going to do a one gene cross. And to do that, we're going to use a 4-box Punnett square. 4-box. Okay? So if we look now, Punnett squares are used to show possible offspring I could get if I cross 2 particular parents. And so we're going to put one parent, the parent alleles go on the top for one parent and on the side for the other parent. And then the boxes, putting those together, show us the possible combinations that I could get in my offspring. So if this parent gives this allele and this parent gives this allele, I could put them together to see one possible combination from these parents would be an offspring that gets big T, big T. Okay, so let's go through the steps. And if you know what the steps are, every Punnett is the same. You set it up the same, and it's going to be easy to work through and do the Punnett problems. So number 1, first you got to figure out what the parent alleles are. That's the hardest part, figuring out the parent alleles. But there's going to be clue words in the Punnett problems to help you figure those out. Once you know what the parent alleles are, you put them in the box, and we're going to do a 4-box Punnett starting out, and then all I got to do is put the combinations together. Once I know what the parent gene choices are, I could figure out what the combinations are going to be. And then all I have to do is fill in the offspring combinations, and then look to see what happened. What kind of phenotypes, what are they going to look like? What kind of genes did they get? What genotypes do the offspring have? And I could count and use math, use probabilities to say what percent are going to look like this, what percent are going to look like that. If you follow those same steps for every Punnett, you will set it up correctly, and it's going to be easy to work through the rest of the problem. Okay, so let's do a couple together. We're going to start, and we're going to make a cross between a tall looking plant and a short. We call them dwarf peas. They're very short looking. And this plant is what we call a pure breeding plant. Always makes tall offspring, always, and their offspring always make tall offspring. And same with this one. This is a short plant. It makes short offspring. Their offspring are going to be short. We would call them pure breeding. Okay, so we're going to use capital T to stand for tall, because tallness is dominant over shortness. And we're going to use, remember, lowercase t, not s, lowercase t for short. And we're going to make a cross. Okay, I'm going to set it up, and I want you to make a cross between a parent plant that's pure tall, and a parent plant that's pure short. A tall plant with a short plant. And when I make a cross, sometimes you'll see it written like this. We put a little kind of an X between. Think I'm going to cross these two things. So if I say pure tall crossed with a pure short, that's what that means. You're making a genetic cross between those two parents. Okay, so first thing, we got to figure out what the parent gene choices are that the parent can give the offspring. And there's clue words now to help you. Remember our letter for tall. We're going to use big T for tall, right? So think tall. I know I got to have at least one big T because my parents are tall, right? And pure tells you something. Remember when we talked about pure? Pure means the same thing as homozygous. I got 2 of the same. Pure means always make offspring that are always going to be tall. That means the parent is going to have 2 big T's. One came from dad, one came from mom. We're going to make them pink and blue. Okay. This is my parent, right? My tall parent plant. In that tall parent plant cells, all of the cells have 2 homologous chromosomes, right, for size. One is carrying the tall gene from dad, one is carrying the tall gene from mom, allele choices, right? So we would say that plant, the other name for pure is homozygous. Remember, homo means the same. So when this organism, when this cell makes gametes, right, makes if this is my dad, make sperm, right? These homologous partners now are going to separate when we do meiosis, and they're going to end up in different, remember, segregation. They're going to go apart and end up in different sperm cells. So big T from this one's going to go one way. The other big T is going to go the other way. Remember, sperm and eggs have one copy of each chromosome, and it's kind of a 50-50 chance of which one goes where, right? But this parent, because it's pure tall, only can give big T choices, right? Big T alleles to its sperm. Okay, let's look at mom. Mom is short, pure short. And so think about, we have clues now. What's our letter choice we're going to use for short? We're going to use little t for short, remember? And pure tells you got to have two of the same. Pure means always gives short choices. So my mom is going to be a little t genotype. That's the genes that she has, right? And we would say that she is also, we could call her homozygous, right? Two of the same gene choices. When that mom plant makes eggs, right, she's going to, one of the homologous choices, or homologous chromosomes is going this way, and the other homologous chromosome partner is going to go the other way. And when I make egg cells now, each egg cell is going to get one of those choices. Remember, sperm and eggs have only one copy of each chromosome. So this parent, because it's pure breeding, only can pass on little t short allele choices to the eggs. Okay, so we figured out what our parent alleles are going to be now. And let's see if we can put them together and see what kind of offspring that these two parents could make. So we're going to pick a Punnett. We're going to use for a one gene cross. We're looking at one characteristic. We're going to make a 4-box Punnett, and I put the parent gene choices. Here's dad up here. Dad could give a big T in this sperm or this big T in this sperm. I put the two choices here. And mom, remember, is little t. She's only got short alleles choices to give. So one goes here, one goes here. Remember, those are the ones that separated when we did meiosis. Each egg cell got one copy of the gene choices. And now I can put them together to figure out what my offspring. If I cross this parent with this parent, what are my possible offspring combinations? So all I have to do is put them together. If dad gives this gene choice and mom gives this gene choice, I'm going to get that combination in my offspring. If dad gives this gene choice and mom gives this gene choice, I could get this combination, or this combination, or this combination. I'm putting them together. This one from here, this one from here, and now those are my possible offspring combinations I could make. What genotypes, what type of genes do the kids get? Each kid is going to get a dominant allele choice, big T from dad, and a recessive allele choice, little t from mom. Big T, little t, big T, little t, big T, little t, big T, little t. Usually when you put the gene combinations together, if you have a dominant one, we put that first, because that's the one that's going to show. Even though this one's here, it's going to be hiding. Remember, that's recessive. So if I put the dominant one first, when I write the letters in, that I can see that right away which one is going to show. So is it wrong that you write it as little t, big T? No, but it's going to be easier to see the answer if you put the dominant one first. So if I look at the genotypes of my offspring, what genotypes, what type of genes do they get? They're all big T, little t. They all have a dominant allele choice. They all have a recessive allele choice. So now I made my combinations. I put my parents in. I looked at what my offspring combinations could be. And now I count and say, what's the chance of getting kids that look this way? What's the chance of getting kids that look that way? So look at the possibilities now. Of this, if this is my offspring, right, it gets a dominant allele, a tall gene, a tall allele, and a short allele, right? Short is recessive. It's not going to show. If that tall allele is there, it's going to hide. This is the one that I'm going to see. The plant is going to look tall, right? even though that recessive one is hiding there. This one is going to look tall. Tallness is dominant. This one is going to look tall. This one's going to look tall. So if I look at phenotype, what are my offspring going to look like? They're all going to look tall, right? Even though they have that little recessive choice there, it's not going to show. So I could do some probabilities. What is the probability, right? If all of my offspring are tall, right, I could do it as a percent. 100% are tall, right? Or how many out of 4, if I counted, how many out of 4 are going to be tall? 4 out of 4 will be tall, right? 4 out of 4 are going to have big T, little t genotype, and 4 out of 4, 100%, are going to look tall. That's how to do a Punnett square. Put the parents in, put the combinations together, and then look to see what's the probability. You can count the boxes. Okay, let's try another one. This time we're going to cross a hybrid parent. And remember, our word hybrid is the same as what other vocab word? Hybrid and heterozygous are the same. Means they have, those words mean the same thing. So if I'm a hybrid or heterozygous, it means I have 2 different allele choices. Dominant, I get the dominant allele, and I also have the recessive allele. Heterozygous and hybrid mean the same thing. So the puzzle, the clue, the Punnett square, the clue word is hybrid. It's telling you what gene choices the parent has. And if I'm hybrid tall, I know I got to have at least one big T. And hybrid tells me I also have a different choice for my second letter, little t. Now, when this parent makes gametes, right, my homologous partners are going to split. One goes one way, one goes the other way. When this parent makes gametes, right, it could give the dominant choice. It could give the recessive choice. It has some different choices to give to the sperm or the egg cells. Gametes are going to have a big T choice or a little t choice. So let's make a cross now. We're going to cross 2 hybrid or heterozygous parents. So hybrid parent can give a big T or it can give a little t. Those are the possible gametes. They go on the top. And my other hybrid parent could give a big T or a little t choice. Hybrid tells me I got to have 2 different choices. Okay, so now let's put our combinations together. If this parent gives big T and this parent gives big T, one possible combination could be an offspring that has 2 dominant alleles, 2 big T's. Also possible, I could give a big T from this parent and a little t from that parent. I could get a big T, little t combination. That's possible, right? I could also get big T, little t from that combination. Or if this parent gives little t and that parent gives little t, I could get a 2 recessive combination, little t, little t. So now look at my offspring. They're not all the same, right? So I could look and count and see what's the probability of getting this one, what's the probability of getting this one by counting offspring in my boxes, right? So if I look now, genotypes, 3 different genotypes, types of genes that my offspring could get. 1 out of 4, count them, 1 out of 4, right, are going to get the big T combination, 1/4, right? These guys. And I have 4 possibilities. So 1 out of 4 are big T. How about these guys though? Big T, little t. 2 out of 4, or I would say 1/2, half of my boxes are going to get the big T, little t, right? Heterozygous combination. And 1 out of 4, these guys are going to get the little t genotype. Okay, those are my genotypes. What types of genes are these guys going to get? Now, phenotypes, I got to count. This one is going to look tall. It's got 2 big T's. It's got 2 dominant allele choices for tallness. This one also is going to look tall. it's got a big T choice, right? Even though that recessive is there, it's not going to show. Remember, big T is dominant. So this one's going to look tall, this one's going to look tall, and this one is going to look tall, right? Phenotype means how's it going to look. So 3 out of 4, these guys are going to look, we would say 75% if I did it as a percent, right? Or 3/4, I could do it as a fraction, right? 3/4 are going to be tall, and this one, 1 out of 4, are going to be short, right? If I did that as a percent, what would that be? 1/4 is 25%. 25% of my offspring are going to be short. 3/4, 75% are going to show the tall trait. Okay, let's practice making gametes, because the hardest part in setting up a punnet is to figure out what the parent gametes are going to be. Once you have those, easy to put them in the boxes, easy to put the combinations together, and then all you got to do is count. So let's practice a little bit. We're going to use big T to stand for tall and little t for short. And we're going to use big R to stand for round seeds, smooth on the surface seeds. And little r is recessive. Wrinkly seeds are recessive to smooth round seeds. Okay? So we're going to use those letters, and we're going to make some parent gametes. And you got to remember, there's clue words to help you do that. Okay, so let's make a cross. And remember, there are clue words now in the problem, in the Punnett problem, to tell you what letters to use. So we're going to start, and we're going to make a cross between a homozygous tall parent. Homozygous tall. There's clue words now to help you remember. So think about tall. What letter we use for tall? I got to have at least one big T. And what does the homozygous part tell you? Homo means, yes, same. They're going to have the same letter. So if I'm a homozygous tall parent, right, I'm going to have a big T choice. And I can tell that because I know I use big T for tall, and homozygous tells me I got to have 2 of the same. If the parent is homozygous for tallness, it's going to have 2 big T allele choices. Now, when I make gametes, remember, separation of my homologous partners. One's going one way, one's going the other way. I'm going to make a big T there, big T there. Remember, gametes get 1 choice, 1 of the allele choices, not 2. Okay, let's try another one. How about a pure wrinkly parent? Remember our letters we talked about, big R is for round, and little r is for wrinkly. So I'm going to use little r, wrinkly. And pure now, remember, pure is telling me, it's a clue word. It's telling me how many letters do I need that are same or different. Pure means the same thing as homozygous. Remember, if I'm pure breeding, I always produce offspring that look the same. So I got to have 2 of the same letters. So if I'm pure and I'm wrinkly, I got to have 2 little r's. I can tell that by the clue words that are here in my problem. Okay, so segregation, 1 goes one way, 1 goes the other way. We're going to make gametes that have little r or little r. Okay, let's try a heterozygous round parent. Remember R, big, we're going to use R for round. Big R stands for round. And what does heterozygous tell me? Heterozygous means the same as hybrid. And if I'm heterozygous, it means I got to have 2 different gene choices, right? That clue word tells me what letters to use in my parent. So big R for round, and if it's heterozygous, I also have to have a little r for wrinkly. Right? Don't see it. I look round, but it's hiding in there. Dominant, round is going to be dominant over wrinkly. Heterozygous means I have 1 of each allele choice. Okay, so now I'm going to make gametes. Separation of homologous partners. One's going one way. Big R goes to one. One of our gametes, right? little r goes the other way, and we're making a sperm or an egg cell that has one of each of the gene choices. Okay, how about a hybrid tall parent? Tall tells me what letter I'm going to use. Big T for tall, right? What does hybrid tell me? Think about our other word that means the same as hybrid. Hybrid is the same as, yes, heterozygous. So I gotta have two different allele choices, right? Hybrid tells me that. Big T, little t. I know I have a big T because I'm tall, but hybrid tells me I also have that recessive choice hiding there that I don't see. Now, making gametes. When this parent cell makes gametes, one of the gametes is going to get the big tall choice, and the other gamete is going to get the recessive or the little short choice. That's all it is to it, making gametes. Now you can get started doing your first set of Punnetts. Don't forget to ask questions and stay tuned for more Punnetts.